So we've been talking quite a bit on how to record EEG, how to get EEG very precisely aligned with our experimental task. So the activity that the participant is doing while we're recording the activity of the brain, so that we can relate what the participant is doing to the activity in the brain. On Monday, Mikkel gave a very nice overview of EEG, already highlighting some of the methods that we use. And one of these methods that we use to study the EEG is looking at event-related potentials, or ERPs, as they're often called. What I want to do in this lecture is so basically to revisit, Mikkel already explained it on Monday. So what is an event-related potential, or what is an evoked potential? And then we're going to discuss what can we use them for, how do they help us to answer the questions. So there's timing and there's amplitude. ERPs have components. ERPs have a specific nomenclature. So we give them like names such as the N1 or the P2. And so what does this 1 and the 2 and the P and the N, what does that mean? If we can get to this, we might also want to go into how do we use the experimental design together with general linear models or with statistical techniques such as analysis of variance or ANOVAs. There are some more advanced methods such as linear mixed model analysis that you could look into, but I think that at this moment we should not be covering that yet. But I do want to highlight how we use ERPs both for the spatial and for the temporal dimension, for the distributions. So the focus is going to be very much again on the upper right corner, so where we do the analysis. But now we're going to use the data that we get from the EEG system very specifically with the timing that we get from the experimental task. If we look at the brain activity that's following a stimulus or an event, then an evoked potential is the activity that we see following a visual, a tactile, or an auditory stimulus. Whereas an event-related potential around any type of event can be considered as like more general. An evoked potential is really an exogenous component. It's something happening external and then the brain responds. So evoked potentials are often sensory. Whereas an event-related potential can follow an external stimulus, but it can also precede something that is happening. So for example, prior to you making a movement, the brain is already active in planning the movement, planning the execution of the movement. So prior to movement onset, we have an event-related potential, which is called the lateralized readiness potential. Event-related potentials can be related to external or exogenous, but also to internal or endogenous activity. Of course, if we have external activity, it is more easy to find them because we can measure or we can sense triggers at the moment that the external event happens. Whereas getting precise timing of the brain doing something itself is more difficult. Evoked potentials, EPs, are often used in clinical research, whereas in cognitive research, in general, we always call it an ERP. Evoked potentials are ERPs. Some of the ERPs are evoked potentials. That's how you can think of them. So what I said is, and what we've been looking at is that we send a trigger, and then related to this trigger, there's a stimulus, and the stimulus induces a certain type of activity in the brain. And if this activity is time-locked, so if we repeat the same experiment multiple times, so if we repeat it over multiple trials, and if the response in the brain is always the same, then we can average and we're going to get this response. EEG is very noisy, so we never get responses like this, because if you get responses like this, you don't need to average. You see them directly. EEG looks much more like this, or in reality, actually, EEG has noise that is way larger than the ERP. But the consequences of the event-related potential having the same deflections over every trial is that by averaging, it is going to stay. Whereas the noise is going to have different amplitudes in every trial. The noise is random over trials, and that means that the noise is going to get smaller and smaller when we average. This is really a very important principle for event-related potentials. There's also activity in the brain which is not time-locked in the same manner. So it might be related to an event, but where the activity shows different deflections. So sometimes it starts with an upgoing deflection, sometimes it starts with a downgoing deflection. If we average this, we're not going to see an averaged event-related potential. So if we're studying this type of activity, we're not using event-related potentials, but we're using time frequency analysis. And if we have time for that, perhaps on Saturday, we could look at time frequency analysis. But in general, I think it's important to first study the most robust methods and the simpler methods. So why do we study ERPs, or why do we study ERP components? So a component already tells that we're thinking of an ERP comprising of multiple elements. So if you present a stimulus, the stimulus goes through the eye, arrives in the brain. What we in fact are thinking of is that the brain is composed of many individual specialized brain areas, where the eye transmits this information, for example, to two brain areas, which then pass it on to a whole cascading network of different modular brain areas that each of them does their own thing. Some might have to do with detecting the color, whereas another brain area might have to do with detecting the brightness, or whether it's moving or not, or whether it's something that you should respond to or not. So what we're doing is, when we're studying ERPs, is we're trying to disentangle the complex pattern of brain activity that follows the stimulus. So if we think about a simple event-related task, then the signal-to-noise ratio, this is also something that Mikkel already mentioned earlier, that is not high enough to observe brain responses in a single attempt or a single trial. So the signal-to-noise ratio is basically how much signal of interest is there compared to how much noise is there. If you would have a signal-to-noise ratio of 1, then the noise would just be as large as the signal of interest. And with a signal-to-noise ratio of 1, well, you can see the event-related potential. If I just go back, for example, if I consider this probably has a signal-to-noise ratio, like each individual trial has probably a signal-to-noise ratio of 10. Like the amplitude here is about 10 times larger than the amplitude of the noise. But what you can see is that by averaging, the noise gets smaller, which means that the signal-to-noise ratio, the signal does not get smaller, the signal does not get larger, the signal stays the same, but the noise gets smaller, so the signal-to-noise ratio gets larger, because I'm dividing by a smaller and smaller number. Since on the single trial the EEG is too noisy, we design a stimulus or task where we repeat the same task or the same stimulus over many trials. For example, every trial every 5 seconds. And if we present a trial every 5 seconds, we can get about 500 trials or 600 trials in 1 hour time. And 1 hour time is a time that we find quite common in an experimental setting, because the participant doesn't mind sitting in the lab for an hour. It takes half an hour to prepare, half an hour to clean up, so the participant is in the lab for about 2 hours. I would also say, if you design an experiment, don't make it too short. Because if you're going to spend half an hour preparing the participant, and if you're only going to record 100 trials, then the participant is done in 10 minutes. Which means that you actually have a very low return on investment, because you spend a lot of time preparing the participant, and you're actually getting little data. So if your participant's there anyway, just make use of the participant. So what we then often do is we just make the experiment long, and the number of trials is often not so much determined by what we need experimentally, but more determined by what can we fit in the available time that we have with the participant. That means that with studies with children, we have few trials. In studies with adults, we have a lot of trials. The experimental manipulation, so we're not interested in the stimulus, we're interested in something subtle happening. For example, what we saw yesterday, like a standard tone versus a deviant tone. So the experimental manipulation is usually a subtle difference between trials. Each trial has a tone, but some of them are standard, some are the oddballs. And the EEG interest is only about 1 second around the stimulus. So that practically means that if we have, in 1 hour, we only get 600 seconds of useful data. So in a way, it's a very inefficient way of recording the data, because there's so much time that we're not using in the analysis. And that's also what we looked at yesterday with the pre-processing, that if you have an hour of data, there's only 600 snippets that are interesting, and that's what you can make use of to make the pre-processing more efficient, so that you don't have to go through a full hour, but that you just go through 600 seconds. Not the topic of today, but actually something that is of relevance. If you record spontaneous or resting state data, that's actually also very often that's done for about 10 minutes. And 10 minutes is also 60 seconds. So it's very common that if we record data from a healthy adult participant, that we're going to use about 10 minutes of data, even though we record data for a full hour. This is how an experimental task would look like. So this is from a widely used study that's used in a lot of tutorials, in which faces were shown. And some of these faces are famous faces, so they're movie stars. Some of these faces are unfamiliar. What you also can see is that there are face-shaped blurry objects, which are not faces, but which have the same physical characteristics as the faces. So this is a sequence of stimuli that is being presented, and you can make different contrasts. So this study was designed such that we can, so in this study 300 pictures were shown. So the first thing that you can do is you can analyze this, like what's the response of the brain to a picture, or to a picture of a face. But there's more detail to this, because there are 100 of these pictures were unfamiliar faces, 100 of these were famous faces, and 100 were actually scrambled faces. So rather than looking what's the response to a picture of a face, you could say, what's the difference between unfamiliar faces versus familiar faces? And there's actually more detail to this study, and this is often how we design studies, because these 300 trials used 50 unfamiliar faces. And these faces were presented for a first time, and these faces were also presented for a second time. Of course, the second time that you see an unfamiliar face, well, it's slightly familiar, because you've just seen it before. Whereas the familiar faces, if you see them for the first time, and if you see them for the second time, well, they're still famous, you'd seen them before. So there's an interaction between these two, and also the scrambled faces were shown twice. In this specific example, there's even more dimensions that we can look at in the analysis. So for example, there's male faces versus female faces. Some of the faces are looking towards the left of the screen. Some of them are looking to the left, some are looking straight, some are looking more towards the right. So there's a lot of details in these faces that you could use in the analysis. This is what's called a factorial design. We have multiple factors, and along these factors we're manipulating stimulus characteristics. So here I'm showing two of the factors that we have. It's the factor of the stimulus that is being presented, which has three categories: unfamiliar, familiar, or scrambled. And it's the factor of the presentation order, first presentation versus second presentation. And then you could consider this to be additional factors, but this is getting too complex. So this is called a factorial design, and we would call this a 3x2 design, because every trial falls into this 3x2 matrix. And with this 3x2 matrix, we could think on how we have analyzed, how we've collected the data. So thinking about this factorial design also helps us to think about the contrast that we can compute, the difference that we can compute. So for example, if we take all of the faces, all of the real faces, so both the familiar and the unfamiliar faces, we compute the mean value here, and we compute the mean value of the scrambled faces. We can look at how is the brain processing faces versus processing a stimulus, a visual stimulus. So this is going to make us sensitive to the face selective area in the brain. Both of them are going to have activity in the visual cortex, but the important difference is that some of the activity in the visual cortex relates to faces, whereas all of the activity in the visual cortex relates to visual input. We can look at the average of the familiar faces versus the average of the unfamiliar faces. Both of them have a face, So that part of the cortical activity is going to be the same. But the memory recollection of the familiar versus the unfamiliar phase is going to be different, because the unfamiliar phases are not stored in memory, whereas the familiar phases are. Or you can look at the first versus the second presentation. At the first presentation, every phase is going to be slightly unfamiliar, even the familiar phase, well, you haven't seen them for some time. Whereas on the second presentation, you think, oh, I just saw that before. So this is more with like short-term memory. This is more with long-term memory. And where it gets interesting with these factorial designs, and that's why we often use them, is if we're going to study the interaction. So if we're going to study how does the response change when we go from the first to the second, and how is this change different between unfamiliar and familiar phases. So this is really what we use ANOVAs for. And these factorial designs, that's a very common strategy that we implement in our experimental tasks. It allows us to look at things that we're interested in, while at the same time suppressing the things that we're not interested in. For example, this allows us to look at phases. If we would put everything together, we would have response to stimuli, but also the response to phases, also the response to familiarity, also the response to short-term memory. So everything together is very unspecific, whereas if we make selective averages and compute the difference between them, we can become selective to very specific elements of our experimental task. So how does this work in slightly more practice? So we have this EEG recording. In this EEG recording, we have triggers. So this is what you've seen yesterday. So we have the triggers indicated here with these vertical lines. And what we do is we cut out trials. So yesterday I've also shown that we cut out these trials, and then we ignore the pieces that are in between the trials. We don't use it. But it's the trials that are precisely timed relative to the stimuli. And that's what we're going to continue with. So these are the trials of the segments. So yesterday we've been processing 50 segments. This afternoon we're going to process 800 segments. And each of these segments, I'm just going to squeeze it together to make them a little bit smaller. Each of these segments is a small matrix with the number of channels times the number of time points in a trial. What we can do is we can stack them after each other. So what you see here is a matrix with numbers, and each matrix is then repeated over many trials. And what we do is we basically average them together. So that's how event-related potentials are computed. So yesterday we've been cutting out these trials, we've been looking at the individual trials, and towards the end of the practical, towards the end of that, like the demonstration that I gave, we were already averaging them and looking at them. Now there's two important characteristics that we can think about. And the first characteristic is that if we look at one row in this matrix, we have a time course. So we have a vector with one channel for all the time points. And if I plot this vector over multiple trials, this is, for example, what I would see. So on each individual trial, I don't really see much activity that relates, or that I can relate to the stimulus that was happening. So what we can do is we can start averaging, as you already explained. If I just take the average of the first trial, so if I just take the first trial, it doesn't change. But if I take the average of 2 trials, or the average of 3 trials, the average of 4 trials, etc., the more trials I average, the smaller the noise is getting. And the more obvious it is getting that the more obvious it's becoming that there's like a late positive response here. That is, well, if you look here, you can see it. But had you only looked at this data, it would have been difficult to distinguish it. So averaging makes brain activity visible that otherwise would not be visible. So this is the principle of event-related potentials. The brain signal of interest is assumed to be constant over trials, give or take that there's different experimental conditions. But let's say that we have one experimental condition, one type of stimulus, then every time the brain activity is the same. And the noise is assumed to be independent over trials. So that's also why the trials are relatively far apart from each other, And that's why we often use a random inter-trial interval. The participant is not predicting the stimulus, doesn't really know where the stimulus happens. It's just like slightly randomized, and that means that the noise is independent. If we then average, the noise in the average is going to be proportional to 1 divided by the square root of the number of trials. So if we average over 2 trials, the square root of 2 is 1.4. So if I average over 2 trials, the noise is going to get 40% less. If I average over 4 trials, the square root of 4 is 2, then the noise is going to get a factor of 2 less. But typically the noise is a factor of 10 times the amount. Typically the signal to noise ratio is about like 1/10. So the noise is 10 times larger than the signal. So with 10 trials, the signal to noise is only 3 times better. With 100 trials, the signal to noise is square root of 100, which means that it's 10 times better. So yesterday Mikkel already said, or the day before yesterday, Mikkel already said, it's quite common to have 100 trials or more. And that is necessary because otherwise we're not going to see the signal. Because the signal is so small compared to the noise, so we need to average a lot because the averaging is actually relatively inefficient because of us taking the square root. Okay. We have the time course, we have the averaging. Now we also can think about like what's happening in the brain and think more about this modularity. So if we have a source in the back of the brain, and let's assume that this source has a relatively low frequency component, then this dipole is going to be visible all over the scalp. It's going to be more visible, so it's going to have a larger amplitude here at the back. It's going to have a smaller amplitude here at the front. And if at the same time we also have a source that is, for example, in the front of the brain, again, this source is going to be visible. It's going to be stronger in the front of the brain. It's going to be weaker in the back. So what happens is that these two sources, these two dipoles, they both have a potential distribution. These potential distributions, they add up. This is just a linear superposition. And as a consequence, an electrode that is in between these two sources is going to see both of them. And it's going to see the sum of the activity of these two. So what you can recognize is that it has both the low frequency characteristics of this one and the high frequency characteristics of this one. Other locations on the scalp might show more of the low frequency source or more of the high frequency source. So if we're thinking about recordings of EEG on the scalp, we're always thinking of a mixture of the underlying source activity, of the underlying activity that we can model as equivalent current dipoles. So we have a linear model for event-related potential activity. And this results in a very important strategy that we use during the analysis. So if the data consists of a signal of interest plus noise, then we have data in condition 1, which is a signal in condition 1 plus noise. And I have the data in condition 2, which is a signal in condition 2 plus the noise. By averaging, so the signal is related to the task, the noise is not related to the task. So that's also why the signal in the two conditions is different. It could be a standard or a deviant, or it could be a phase versus a scrambled phase. If we look at the ERP is the average of the data in condition 1, or the ERP is the average in data in condition 2. The signal is assumed to be the same on every trial. So if I average something that is the same, I'm just going to get the same thing. So the signal does not change due to the averaging, but it's the noise that gets smaller. Now the important strategy that we use is that we can look at the difference between these two potentials. So if we subtract ERP1 and ERP2, we're looking at signal 1 minus signal 2. And then of course there's still going to be some noise. So this is the strategy that we use in these factorial designs. Because if the signal, for example, has visual cortex activity due to a blurry blob, and it has visual activity due to a face, then that part of the activity is going to be the same in the two conditions, and we're going to subtract it. And the only difference that I have between signal 1 and signal 2 in the example that I showed before is due to the stimulus being a face. It's not due to the stimulus being visual, but it's due to the stimulus being a face. So by subtracting the ERPs that we compute in both conditions, we can make it selective to facial processing and thereby remove the visual processing. Is this clear? Yeah? Okay, so an event-related potential can consist of multiple components. And these multiple components are thought to respond differently to the different aspects of the experimental stimulus. So for example, if we have an auditory oddball task, it's something that we demonstrated. It's like beep, 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 boop, beep, boop, etc. Where the boops are the irregular ones. What we can do, if we want to be really careful about a task, we can also make a task that's boop, 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 beep, boop, 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 beep. What we're then doing is we're making the task balanced. So we have a beep, and the beep is sometimes expected in the first half of the experiment, and in the second half of the experiment, the beep is unexpected. So just ignoring the boops, we can look at the cortical response to the beep when it's expected and the cortical response to the beep when it's unexpected. The auditory cortex, so the primary auditory cortex, is going to respond pretty much the same because, well, a beep is a beep. But the expectancy, of course, is very different. So using these type of experimentally designed tasks where we have one of the features being the stimulus, one of the factors being the stimulus, the other factor being whether it's expected or unexpected, so this is a 2x2 experimental task. It allows us to separate the sensory activity from the expectancy. So the sensory input of the beep is always processed by the auditory cortex in the same way, but the two conditions have a different surprise response. So this is where we would use like an ANOVA to get this factorial design so that we can look at the expected stimuli minus or the unexpected stimuli minus the expected stimuli, and then we can zoom in on the brain region that is responsible for expectancy and ignore the brain regions that are responsible for auditory processing. So in this case, it's actually the primary auditory cortex that is already responding to the expected or the unexpected stimulus. So the brain region for expected sounds is very close to the brain region for sound processing in general. So this experiment is called an auditory oddball experiment, and it elicits a mismatch negativity. The mismatch negativity is not the sensory response to the beep or to the boop, but it's the difference in the standard expected versus the deviant or the unexpected stimulus. But the mismatch negativity is also, so if the beeps are unexpected and the boops are expected, then the mismatch negative is the difference between them. But the mismatch negative is also the difference the other way around, depending on which one is expected and which one is unexpected. So this allows us to use all the trials that we have collected and to sort them in expected minus unexpected. And that allows us to look at the brain region that is related to processing of expectancy in auditory responses. And this mismatch negativity is an activity which is like localized, which can be localized using dipole fitting methods in the auditory cortex, and it's active around 150 to 200 milliseconds following stimulus onset. The auditory processing starts already earlier, but the detection, whether it's expected or unexpected, is a little bit later. This is very similar, like this is another example. So this is what if we present faces versus houses. It's a very common task, like a lot of people have looked at this. A face, like is animate, you recognize them, there's like emotional processing involved. A house, well, you also recognize, but it has very different structure and has very different meaning towards the participant. You can do the same thing with cars or flowers or scrambled faces. So you can do faces versus scrambled faces, faces versus cars. Faces are processed specially because we're so much tuned to other people's facial expressions. And if you do such a task where you contrast faces versus visual stimuli that are similar but at another face, you get a component that is called the N170 component, which originates from the fusiform face area, which is located on the ventral surface of the occipital pole. So this is a spatial topography of the N170 component, where you should think of this being a view from the back of the brain. It's basically like this. And what you see is that there's activity here on the lower side on both hemispheres, and that is the negative activity, and that activity is strong 170 milliseconds after the presentation of the face and after the presentation of the house. So this difference, that's basically the N170. And what you also see very clearly here, so this is the example with faces versus faces versus cars, is that the earlier activity on these electrodes, the so-called P1 component, is not really distinguishing. So the primary visual cortex is just processing the stimulus in the same way, but a little bit later in time, the brain starts processing the stimuli differently because the fusiform face area becomes involved and the facial recognition kicks in. What I've now done is I've shown you some examples. So we have this mismatch negativity, we had the P1 components, occipital, we had this N170 component, and these are all components that have a specific nomenclature that's important that you're aware of. So we often have a P followed by something or an N followed by something. So for example, a P1 or a P3. The N or the P refers to whether the component is positive or negative on the electrodes on which it is reported. So an N1 is a negative component. And the number that follows indicates which component it is. So here, this is the first positive component, so that's why it's called a P1. You could say, well, this is the second component, and it's the first negative component. So, but what we do here is actually we're not looking at the component, we're looking at the difference, and the difference that gets the name N170 because it is maximal at 170 milliseconds following stimulus onset. So this is where the second type of terminology comes in. So we have P followed by a number in milliseconds, or N followed by a number of milliseconds. So for example, you have the P50 or the N170. Again, P or N refers to it being a positive or a negative deflection, but the number is now the latency, approximately in milliseconds. So the P300, that's a very well-known component. You get it in as a reaction to surprising stimuli. It's a positive deflection. It's largely positive here over C3, over CZ, so over central brain areas, and it's around 300 milliseconds following stimuli. And sometimes we have components that have more unique names. So for example, the readiness potential. The readiness potential was discovered by a German researcher, and back then in Germany they were still publishing everything in German. So it's also known as the Bereitschaftspotential. And this Bereitschaftspotential is still terminology that some people stick to, although I think the tendency is now more to use the English term, which is the readiness potential. The readiness potential is activity that you get prior to making a response. Okay, so prior to making a response, there's planning involved, and part of that planning is general, and part of that planning has to do with the execution of the motor task. So what you can do is you can design an experiment in which participants prepare a movement with the left hand, or where they prepare a movement with the right hand. And if you subtract those two conditions, the general planning, which is the same for the left and right hand, cancels out, and then you get a lateralized readiness potential, where the left hand planning is executed by right motor cortex and right hand planning is executed by left motor cortex. So these are the tricks that we use to get these components. A CNV component is a contingent negative variation. It's also one of these components that is related to the planning. It's a very slow building negative component prior to, for example, making a decision. or a very specific component, like the N2PC. It's the second negative component, which is posterior and contralateral to the stimulus. It's something that happens over here, typically around 200 milliseconds. Or like, sorry, if the stimulus is here, it happens here. If the stimulus is here, it happens here. It's like it's contralateral to the stimulus. So there's a whole range of these components, and like knowing these components really helps, because it means that if you see one of these components, you're going to recognize it, you can link it to existing knowledge about this component. And Steve Luck is like one of the really well-known EEG and ERP researchers. He wrote a book in which is an introduction to event potential technique. And Mikkel actually brought the book with him. So, and he also took it from his hotel room. So if you just look around, Mikkel has the book with him. So feel free to have a look in the book and see whether you want to like buy the book or find a PDF version online, which is probably also possible, so that you can read through the book and learn more about ERP components. Okay, so what I've shown is how do we compute ERP components. And there are two aspects to ERP components that I've now been highlighting. The first is, when does the activity start? But actually what we more often look at is like, when is the activity different between two conditions? Like visual activity or sensory activity, auditory activity, we already know how that works. Has been studied for decades. What is interesting is like, how is this activity going to be transferred to higher order brain areas where the important differences in cognitive processing happen. So often we ask the question, when is the activity different between conditions? And if we look at the when question, we're looking at timing. So we're looking at how the activity evolves over time. A second question that we can ask is, if we look at all the channels at one time point, then we can ask the question, what is the topographic distribution in one condition at a certain latency? So if we present a visual stimulus, visual cortex is going to process it. So we're going to have a P1 component over the occipital area. But very often what we're interested in is, what is the topographic distribution of the difference at a certain latency. And that's, for example, what we see with the faces, which are processed here, versus the houses, which are processed here. But there is a subtle difference, 170 milliseconds after stimulus presentation, and that difference can be localized to the fusiform face area, which is a specific area related to face processing. Oh yeah, and by the way, perhaps it's also relevant, we're not only studying ERPs, we're also studying other things. So if you get a brain lesion, specifically in that area, then patients are unable to do facial recognition. Patients become unable to do emotion recognition. So we know from converging evidence from very different types of research, we know that this specific area in the brain is the area where faces are being processed. So it's not only EEG research, but for example also lesion research, it's also fMRI research, etc. But it's the topography that helps us to determine the location of the activity or the location of the difference in activity. And I was planning tomorrow morning that we would do a lecture on source localization, like localizing where in the brain activity is, given that we have spatial topography. So by studying ERP components, we're studying the when activity happens and where activity happens. We have different functions in different brain areas, and this brain, we really think of this as a hierarchical functional network. At the same time, we have serial sequential processing, and we have parallel processing at different times, and also in different areas. And by using the subtraction technique of ERP components, and by looking at the components, the latency and the topography of the ERP components, we basically disentangle the functional network, and we try to zoom in, for example, in a specific area that is responsible for a specific type of processing. So this is very much how a lot of the EEG research is done. It's basically by using ERPs and by looking at subtraction waves. If you want to make it really special, so this is the cortical hierarchy of the visual cortex. So this is not studied in humans, at least back then it was not studied in humans. This was studied with invasive recordings in macaque monkeys by David van Esten and Velleman. And this is a very influential one. And what you can do is if you go invasive, you can get these very detailed maps. But these maps are now also being confirmed using fMRI, but also using EEG techniques. And by looking at the specific hierarchical structure of how information is flowing from one to the other region, we're able to zoom in on very specific brain regions and to study their function. And of course, we can also study their function in relation to what the participant is doing, or for example, to the relation of the condition that the patient has. So this is also relevant for patient research. And that helps us to disentangle activity in the brain.